Microplastics are ubiquitous component of the environment. According to National Ocean Service, microplastics are plastic pieces with a size of 1 to 5 millimeters in length. Microplastics are classified as either primary or secondary. Primary microplastics are synthetic polymer used in industrial application, mainly as exfoliate for chemical formulation and the manufacture of clothing. Microbeads are a category of primary microplastics that are less than 2 millimeters thick and are used in healthcare and cosmetic products. Secondary microplastics develop out of the disintegration of large plastic under various degradation process. The source of microplastics include industrial process, domestic activities, and the natural degradation of plastic in the environment. Microplastics are present in water bodies, soil, air, food, and drinking water. Microplastic poses health risk to humans when ingested in significant quantity in food, water, or air. However, there is no scientific data on amounts that can cause toxic effects. The particle can get into our food and water through plastic containers, contaminated water source, and the food chain. Humans can also ingest microplastic present in the air through breathing. Pollution, disposal of wastewater, industrial influence, surface runoff, and atmospheric deposition contribute to a presence of microplastic in water bodies. The effect of microplastics on human health is still a subject of research. Preliminary studies indicate a variety of effects such as obesity, reproductive issues, delayed development in children, and organ problems. The effects are derived from the known health impact of the chemical and metal substance present in microplastic. Recent research estimates indicate that a person consumes over 74,000 microplastics annually, while other studies indicate a weekly consumption of up to 5 grams. The quantity are alarming, considering the rising level of pollution in urban areas. Air pollution poses a great health risk to cities in the developing nation. A study by Schnitt Mercer about the level of particulate concentration along cycle paths in Berlin established that a person cycling along the main highway is exposed to an average of 7,020 particles per cubic centimeter. The implication is that urban dwellers are exposed to high level of microplastics. The high concentration are usually at the source, but the particles get distributed to different environments through the air, water, and human activities. Microplastic poses health risk through their physical presence in human bodies, chemical composition, and attached microorganism called biofilm. Bacteria biofilm poses a huge threat to health because they are resistant to antibiotic treatment regime, resistant to disinfectant chemicals, physical operation, and the immune system. Biofilm on microplastic can be a potent method of disease transmission. The bacteria colony in a biofilm can share resources and communicate with each other through a process called quorum sensing, which involves the sharing of chemical signal molecules. Quorum sensing enables the microorganism colony to analyze the environment and adjust its behavior for increased survival. The level of adaptability and its wide range of infectious microorganisms that can attach to microplastic present a major threat to human health. Other microorganisms that form biofilm include protists and fungi. Microplastics are therefore potential carriers of disease-causing organisms. The size, shape, hydrophobicity of the particles also influences the range of the harm they can cause in the body. The particles cause physical operation on gut tissue as well as during translocation to other parts of the body. Physical operation can cause inflammation or broken tissues, allowing the entry of microorganisms into the body. Some microplastics may contain chemicals that get into the human body and cause health complications. According to Campanile, microplastics contain two major categories of chemicals. The first category includes additives and polymer raw materials, while the second category is the chemical absorbed from the environment during dispersal. The additives are used to add quality to plastics, and they include antioxidants, dyes, flame retardants, lubricants, and plasticizers. Some of the toxic chemicals include bisphenol A, phthalates, brominated flame retardants, and styrene. The leaching of bisphenol A into human body can interfere with hormonal functioning, leading to reduced fertility, obesity, and cardiovascular disease. Phthalates can disrupt hormones and cause low testosterone level in male infants. Styrene can cause nervous system disorder and cancer and hearing loss. Brominated flame retardants disrupt the endocrine system and affect the functioning of hormone-receptive organs. The effects of such disruption include metabolic disorder, asthma, and hormone cancers. Polychlorinated biphenyl can weaken the immune system and affect reproduction. Wright and Cayley explained that microplastic release in the textile industry can cause respiratory infection after prolonged exposure. Microplastics have deleterious effects on animals. Marine animals ingest the particles through feeding and breathing underwater. The chemicals present in the microplastic pose serious health risk to the animals. Endocrine disruptors like phthalates lead to reproductive problems, gastrointestinal irritation, changes in fish behavior, and interference with lipid metabolism. Smith explains that accumulation of microplastic in body can have physical effects on terrestrial and marine animals. The effects of the accumulation include accumulation of tissue, necrosis, and cellular proliferation. Ingestion of the microplastic reduces respiratory function in blue crab and the formulation of granulomas in blue mussel. According to Wright and Cayley, microplastic granules can cause embolization of small vessels in animal bodies. The long-term accumulation of particles in animal body can trigger oxidative stress, genotoxicity, and apoptosis. Long-term effects include fibrosis, carcinogenesis, and tissue damage. Dewis and Coors explains that high concentration of microplastics have negative physical effects on marine animals. High uptake of the particles lead to reducing feeding and subsequently lead to lower energy level and reduced physical functions. According to Martin and Gaffarino, microplastic induce an artificial sense of sensation in the gut leading to reduced feeding, physical injuries on gut cells, and decreased absorption of food. A study by Martin and Gaffarino on the Dolphinia magda straw species found that prolonged exposure to microplastics lead to a generational decline. Fish that consumed microplastics produced fewer offspring up to the third generation. The offspring also had a higher mortality rate, indicating reduced fitness. Recovery to normal productivity level after exposure to the particle took a longer period, covering multiple generations. Ingested microplastic can be translocated into the tissue and cells of animals. According to Carman, Shallow, and Unwin, the translocation of microplastic from the gut into lymphatic system of rodent causes thrombosis and cellular damage. Biomagnification also occurs through food webs. As a result, a significant portion of the microplastic consumed by marine life ends up in the body of fish-eating birds as well as other aquatic organisms. Similarly, microplastics consumed by fish and crustaceans often end up in the human body through consumption. Although the human body can excrete significant quantity of particles, some microplastics can translocate from the gut into the lymph nodes and the blood circulatory system.